The topic of this talk today is actually a part of Anatolian Lynx project in Turkey. So it's the conservation, ecology, and genetics of Eurasian lynx in Anatolia. Pardon, geçemedim. Şöyle mi yok? Evet. So, as we all know, large carnivores are often flagship or umbrella species for conservation of biodiversity. And by conserving them, actually, we are not only conserving the species themselves, but also a wide array of other species, because when we dedicate protection areas for large carnivores, such as here in India, you see there are 50 tiger reserves. We also protect many other species together with them. Their prey, from insects to birds to plant species, everything in those protected areas. And also here you can see that the 20 dedicated snow leopard reserves in Central Asia, and they cover actually huge areas where we not only conserve the snow leopards, but also many other species together with them. But in order to conserve large carnivores or any species, we actually need a lot of information. I press admit, okay.
Deniz, Deniz pardon, biraz sesin takılıyor. İstersen videonu kapatalım mı?
Olur,
arada
tabii. Şöyle kapattım.
Tamamdır.
So in order to conserve any species, we need baseline information such as their dietary requirements, their population status, not only them, but also their prey, their genetic status, spatial behavior, their habitat requirements, the levels and causes of conflict, if there are any, and also the threats actually threatening the populations themselves. I couldn't pass again. Okay. So what is the status of large carnivore research and conservation in Turkey? Actually, many species missing baseline ecological and genetic data. And here you can see the list of our large carnivores, starting with red fox. This was the threshold actually I took as large carnivore. Some studies say that red foxes are large carnivores, and some others that they are smaller carnivore species. As you see, for Turkey, we only have some species for which the IUCN assessment have been done. These are the brown bear, the wolf, the golden jackal, and the red fox, all of which actually have least concern states assessments, but actually some little populations of brown bear has actually critically endangered status, such as southwestern Turkish populations, where there are only few remnant individuals existing. And the rest of them, most of the carnivore species actually not evaluated still in Turkey. And when we look at the research done on these species, actually the brown bear and the lynx now are the luckiest ones, but for other species we have only several studies. And for jungle cat and striped hyena, actually, we have only 2 published papers, and maybe 3 now. I don't know. This was from last year. But yeah, we are lacking lots of research in Turkey on large carnivores. And these are the extinct species that are already gone. There's a funny thing here, actually. Although we have the least conducted research on these 2 species, the striped hyena and the jungle cat, one of them has a wildlife reserve dedicated to conserve it, and the other one has a conservation action plan. But actually, we know that we didn't have enough data even to make a conservation action plan on the species. For example, if you look at this was the conservation action plan of jungle cat in Adana. When you look at this conservation action plan, actually, you will see that lots of data is missing in order to actually make a good conservation action plan as well. For example, it's stated that the species having 9 kittens in the bird, which is totally wrong. So we need to do lots of research. And why can Eurasian lynx can be a good model actually for a conservation action plan or large carnivore conservation in Turkey? because actually this species has a wide global and local distribution. You can find it in most of the mountain ranges in Turkey. The subspecies occurring in Turkey, the Caucasian lynx, actually has a very different ecology than other lynx populations. So it gives us the opportunity actually to study the same species in a different locality, in a different geography, and try to understand its evolutionary ecology and also genetics, maybe. It can occur with very high densities in comparison to Europe in Turkey. Its habitat is currently under threat. There are hydropower plants. There are many different, mining activities, and also, let's say, the wind turbines and many different constructions being done in their habitat. So it's a good time also for doing it. And Anatolia is very important for this subspecies because almost 70% of its population is only occurring in Anatolia. Although it's shown in Iran, it has a high distribution, wide distribution. It's in very low density because it co-occurs with leopards, and leopards suppress Eurasian lynx populations there. So it's mainly in Caucasus, Turkey, and some in Iran. So in the context of Anatolian Lynx project, actually, I tried to assess its foraging ecology, its conservation genetics, spatial behavior, like the home range size and density. And also, lately, actually, after my PhD, we also published research on its phylogeography, its evolutionary history as well, which in common minutes, actually, I will give information about these, let's say, these chapters. So I selected 3 different study areas in Turkey with my other colleagues. One of them, the forest steppe ecosystem in northwestern Turkey, was my focal study area where I did the long-term population monitoring and the research that needed long-term monitoring, actually, such as the spatial behavior, where you needed to capture the lynx, put GPS collars, or repeatedly collect samples to do conservation genetic studies, and also for its density. But from other areas, we also collected, for example, the dietary data to assess the dietary ecology of different populations. And this was also used for phylogeographic study to see the genetic background of different populations in Turkey and also their connection to other populations. So the foraging ecology, actually it's known that it was a generalization of Eurasian lynx always preying on ungulates. And this was mostly coming, of course, from the studies in Europe, dietary studies conducted in Europe, which indicated that roe deer was the main prey of Eurasian lynx, and Eurasian lynx is a ungulate specialist lynx species, which generally preys on roe deer, shama, other deer species in a whole lynx distribution. And although we have some suitable preys in Turkey, such as the roe deer, wild goat, other ungulates such as shama, wild sheep, and deer species, it only co-occurs with wild boar and brown hare in its whole distribution in Turkey. And at different study areas, it also has some, let's say, co-occurrence with other ungulate species. However, we know that, for example, lynx in Turkey and roe deer distribution, they are not overlapping much. For example, the Black Sea mountains and the deciduous forest there, although they harbor roe deer, lynx doesn't exist in these habitats. So then this brings some hypothesis and hypothesis testing. So the null hypothesis here was Asian populations of Eurasian lynx are also ungulate specialists as European Eurasian lynx populations, and only feed on lagomorphs when large prey is not available. Like, for example, in Turkey, we have lagomorphs everywhere, and ungulate species have limited distribution. Maybe because of that, they are preying on lagomorphs. This was the idea. But actually, there is also an alternative hypothesis which can state that Eurasian lynx first evolved in Asia and also started as a logomorph specialist like other 3 lynx species you see here. For example, the Iberian lynx, the first one is a rabbit specialist lynx. The lynx, the bobcat, is also a predator of small prey species. And you know the famous Canadian lynx and snowshoe hare predator-prey relationship, which is also a subject of many ecology books. But only Eurasian lynx has bigger size, and it was classified as the ungulate specialist. But actually, it's also the oldest lynx species. And as other lynx species has also logomorph specialization, we can also maybe say that some Eurasian lynx populations in Asia can still be logomorph specialists, and maybe the species also started as a logomorph specialist. So we wanted to test this. In order to test this, we had 3 different study areas where actually lynx was. In Mediterranean study area, it was sympatric with wild goat. In forest steppe, it was sympatric with red deer. And in subalpine study area, it was sympatric with wild goat and chamois together. So this gave us a good opportunity to study the dietary preferences of lynx co-occurred with ungulate species, but also with brown hare. We collected scat samples in these study areas. We calculated and estimated the consumed biomass of each prey species in lynx diet. We calculated the prey densities using random encounter models from camera trapping data. And later on, we quantified the Chessen's selectivity index for the prey preferences. And also, in the end, we actually compared the functional response of lynx populations in Turkey together with other lynx populations in Europe. And also we added Canadian lynx and Iberian lynx to this equation to see actually which lynx population better explains Eurasian lynx ecology in Turkey. I mean, like, is it a logomorph specialist lynx or is it an ungulate specialist, to see in a figure altogether? You see some, actually, yeah, this is the prey species under microscope. When you do dietary analysis and microscopic analysis, these are the medulla structure of each prey species under the microscope, so that you can identify each prey like this. And then the results showed us that actually Eurasian lynx in Turkey, all 3 populations had the brown hare as the main prey species. And then later on, ungulates also contributed a little bit in 2 study areas in the Mediterranean and forest steppe. But also there was a contribution of some wild goats and some domestic goat in subalpine Artvin study area. But carnivores were also contributing a lot to the lynx diet in the northern study areas. And the rest of the prey species were not so important in lynx diet, actually. We also found some proof for the cannibalism in lynx diet, which was also surprising. And then we also thought that maybe it was related to the very high lynx density in these study areas, because populations, some predator populations actually use intraspecific killing, I mean, killing of the same species. And once they kill them, maybe they also eat them. Here you see the results of random encounter model, I mean, density estimates in 3 different study areas for brown hare in all study areas, and also the sympatric ungulate species. For example, in Mediterranean and forest steppe study areas, the brown hare had a high density, but the other populations, they were in lower densities, of course. But in subalpine study area, we have very low amount of brown hare present in the study area. Although it was like this, brown hare was 75% found in the lynx diet, actually, which can also be an indication of very high, you see the Chessen selectivity index here, which also, lynx had very high preference towards brown hare in the study area. But in all study areas, the main lynx prey and the main preference was towards the brown hare. So you can see we used here the biomass in diet for 3 different study areas for all species. the biomass available in the ecosystem, which we calculated from the prey densities, and then we calculated the Chesson selectivity index, and we got the preference results. And here you can see the functional response results, actually, the quantification of the functional response for each species. Raw deer for European Eurasian lynx populations, we had 7 different lynx populations here. Here you see the prey density per square kilometer and prey intake per predator per day, actually. And there was a one lagomorph eating lynx population, Eurasian lynx population in Finland, because this population actually, there were no ungulate species in the study area. They had to prey on mountain hares. Here you see Anatolian, 3 Anatolian lynx populations, our populations, I mean. Here, this is the Canadian lynx and the Iberian lynx. So as you see, they are much closer to the hare specialist lynx populations and species. And as you see, we also calculated the prey intake per day for each population, and lynx in Anatolia was much closer to Canadian lynx and Iberian lynx in terms of their prey intake per day, rather than the European lynx, which also actually explains why they are much smaller than the European lynx, because our lynx individuals in Turkey are, let's say, 2/3 of the lynx individuals in Europe as body mass. For example, the males are 16 kilos, females are 13 kilos in Turkey, but this can be 20 kilos for females in Europe and 30 kilos for males. So our lynx are also much smaller, and they have this adaptation for local diet. So our conclusions were Eurasian lynx as a species is not an ungulate specialist. So there are also some populations which can be specialized on lagomorphs. Lagomorph dominated diet, smaller body size, lower prey intake are evidences of specialization on lagomorph in Turkey, let's say. And ungulate-based diet in Europe might be a recent adjustment to the high densities of ungulates in Europe, actually, because after the eradication of carnivore species, many carnivore populations in Europe in the last 200 years, of ungulate populations there. So maybe that's why the ungulate-based diet in Europe is a result of this in Europe. As I said, there was cannibalism. And the main conclusion was actually we cannot generalize findings of local studies, for example, in Europe, to whole species distribution, because when you have a wide-ranging carnivore population or species, such as in this case, Eurasian lynx in Europe, Asia, Central Asia, Far East, and so on, you also have a lot of diversity in feeding ecology among different populations. And lagomorph prey should become an essential component of conservation activities in Europe. Emrah, am I going too slow? Because I have many things to tell. Or is it okay? Cool. So the second study was about the population genetics. This is the focal study area in northwestern Turkey. And here you see my study area where I collected many scat samples, but also I had some traps to catch lynx to put GPS collars. So I used 2 different types of sampling. One of them was scat sampling, non-invasive sampling, and the other one was trapping lynx and collecting some tissue samples from them. And here my idea was actually sampling type does not influence the measured genetic diversity. This was the null hypothesis. And the alternative hypothesis was sampling type is important, and depending on animal behavior, can influence diversity estimates significantly, because we know that actually lynx populations or any carnivore population is not distributed randomly in the landscape. Because we have, for example, 7 female individuals in the study area, and we have 4 male individuals, but among the 5 female individuals from camera trapping, actually, I knew that some of them were actually related to each other. For example, a mother here and the daughter here. So from the beginning, you can actually expect that maybe you will collect some related Lynx data when you capture them. Anyways, I will tell you more about this later. So I trained my dog, which is Gürbüz, you see here, for collecting scat samples. And for this study, we used 14 microsatellite loci and 2 sexing loci to identify the Lynx individuals and the genotypes. 11 of these loci were successful. 2 of them deviated from Heidi Weinberg, that's why they were removed. And 1 locus was monomorphic, so it couldn't tell anything about the individuals, so we removed it as well. And then we also used 8 loci to increase genotype numbers, because with 11 genotypes, actually we couldn't get as much individuals as we expected, because most of these samples were scat samples, and I used swabs to collect the samples. Some of them aged, let's say, 5 days. The others, they were like 30 days old. So we also used 8 loci to conduct some analysis. Here you see the effort per different, let's say, study type. Non-invasively collected samples, invasively collected samples. So I conducted 961 trapping days to catch lynx, but I did only 52 days to collect scat samples, which was much less effort, and I collected 171 samples with these days, actually. And when we, depending on the loci numbers, we had, for example, 27 genotypes per 11 loci. This was 45 for 8 loci by non-invasive sampling. And for invasive, it was 11, 12. Some of these lynx were territorials. Some of them were kittens. And there were also some dispersers and floaters. And as you can see from non-invasively collected samples, we had higher numbers of each type of lynx individual, but also we could catch more, for example, disperser floater lynx individuals, which could increase the general genetic diversity measures, let's say, estimated in the study. And when we compared this lynx population in terms of its genetic diversity measures, it was one of the most genetically diverse population compared to other European populations. As you see here, for example, the allelic richness, expected and observed heterozygosity were among the highest. And we couldn't find any sign of inbreeding in this population. And although these lynx individuals, actually, most of the females were highly related to each other, still we had a low mean relatedness of the population. Here you see the reintroduced populations, they had the lowest, let's say, genetic diversity measures, lowest genetic diversity, because actually most of these populations were established by only few lynx individuals. For example, in Bavaria, let's say, there were, I think, around 10 or 12 individuals released. In most of those populations, these reintroduced populations actually were started few individuals, and some of these individuals were related to each other. Here you can see the relatedness of individuals inside the population to each other. This is Anatolia, my study site, which had the lowest relatedness values, and together with Latvia and Russia. These were the 3 populations which had the lowest relatedness inside the population, although my study area was only 400 square kilometers and most of the individuals were related to each other. And for example, in Russia, in Kirov region, the samples were collected in much higher study, like much wider study area. And this little population actually had the same low relatedness like that population. And here you can see the all reintroduced populations together with an isolated population in Slovakia and Poland. They were all actually highly related to each each other. Although this is, for example, an autochthonous population in Poland, they already have some evidence of inbreeding inside the population. And when we came actually to test our hypothesis, to test the success of sampling type actually in catching the population genetic diversity, here you see the triangles are all samples assessed together. Let's say the invasive and non-invasive samples. And you see the genetic diversity, I mean, how it increases with number of samples. For example, the mean number of alleles and expected heterozygosity, two measures. And here you see the non-invasive sampling only, the blue one. Actually, it's very close to, let's say, whole gene pool be collected, whole samples together. I mean, non-invasive and invasive samples together. Actually, by only collecting non-invasive samples, we could get a result very close to non-invasive and invasive sampling together. So then you can ask, for example, do I need to collect any invasive samples to assess conservation genetics of a carnivore population? So we have seen the significant difference between the non-invasive and invasive sampling in terms of explaining the genetic diversity inside the population. As you see, invasive sampling underestimates the genetic diversity because, actually, I should also tell this, because when you install traps to catch lynx inside their territories, you mostly catch the territorial lynx individual inside that study, I mean, in that location, or you can catch the kitten, or let's say the young of that individual, let's say 1-year-old sibling, not sibling, but offspring of that individual. And because females, neighboring females are related to each other, for example, their mothers and daughters, the samples you will get are mostly related to each other, and they are not randomly distributed inside the population. However, by collecting non-invasive samples, you, of course, also collect samples from the territorial individuals, but you also collect samples from the individuals roaming in the study area. For example, individuals coming from other study areas, other territories. So non-invasive sampling is always increasing the genetic diversity measures when you want to conduct a conservation genetic study for territorial species, of course. And this is because the population is not evenly sampled by the invasive sampling. And of course, the genetic relatedness of females was much higher than the males because males are dispersing in much longer distances. They were relatives, the females. There was philopatry, and we found negative autocorrelation peaking at every second female territory, which means the neighbor of the lynx, female lynx, is her relative, but the second neighbor is not related anymore. Anyways, this is too much detail because I will give a lot of other. I will talk about other things as well. And actually, in this study, I also tried to test if morphology can tell about the relatedness of female individuals. Let's say here you see the mother-daughter pairs. This is İpek, her daughter Asi, her daughter Zeyna, mother, grandmother, daughter relationship is here as well. The MXY is the amount of relatedness between the individuals. And there are two family groups in the study area. One of them is İpek and her daughter and granddaughter, and the other one is Eylül and then her family. There is very little relatedness among these two families, but they are highly related to each other inside the family, let's say. But we couldn't see, for example, the morphology is, for example, we had small dots, a little bit bigger dots, and big spots in a family group. And in another family group, they were also all present which could not tell anything about the relatedness. Actually, when you see, for example, big spots, you cannot say that maybe this female is related with this one, but actually the genetic markers we used, microsatellites, cannot tell about the morphologic patterns between the lynx individuals. This was another result. So we had the conclusions that Anatolian lynx had, the lynx population here had a high genetic diversity, no evidence of inbreeding. Let's say the animal behavior, for example, territoriality and female philopatry can affect the genetic measures, conservation genetic measures, and also should be considered while, let's say, planning a methodology. And this was the first baseline population genetics study for this subspecies, actually, and can be used for other Caucasian lynx populations in Turkey and also in its whole distribution in surrounding countries. Spatial behavior and density. Here, I also had hypothesis. Should I go into detail? Do we have time? Because I have one more chapter later on.
So we have around 25 minutes. I think 10 minutes will be good. Then we will have some time for questions.
Yeah. Okay. Then I will go. Let's say, okay. Let's talk about them briefly. There was a hypothesis by Scandinavian people that the ecosystem productivity and prey base are the key factors determining the home range size in the Eurasian lynx. So, which means higher ecosystem productivity leads to smaller home range size in Eurasian lynx. And I had an alternative hypothesis for this because the study area I'm working in actually is not super high productivity study area, most of which is kind of steppe ecosystem. This is a study area which is in between the steppe and forest ecosystem, and the forest type is very xeric, I mean very dry forest. That's why you cannot expect, for example, to see very high densities of ungulates, but maybe xeric condition adapted species such as logomorphs can tell something about this. Here I colored 3 adult males, 3 adult females, 3 subadults, and fitted them with GPS colors. And then I calculated the kernel and MCP home range sizes for different individuals and categories. And I also estimated the density by using spatial recapture models using 17 camera trap stations. Here you see the kernel and MCP average home range size per category. Here, territorial females. One of the females had actually 2 home ranges because it was monitored for 2 years. Territorial males, non-territorial young males, and actually in this study, for the first time, we found out that lynx, if there is very high density and if there is not enough space in the cape, there are also non-territorial adult males which can roam in huge home ranges. For example, one of these males was roaming in 4,000 square kilometers. When you compare this to a female home range, which is only 50 square kilometers, is something really huge. He was roaming the same area and trying to find a territory, empty territory where there's a female, but he was not successful during the study. So, which also led us to the floater adult male concept, which was already found for cheetahs, African cheetahs, by my colleague in my institute, actually. There are also some cheetahs like this roaming in huge areas until they find an empty territory. Territorial females, males, you can see the average home range size difference with floater males, these adult males without territories roaming huge areas. And here territorial males only 3 times bigger than the territorial female home range size. Here are the home ranges of different individuals. As you see, these are the females, these are the territorial males. This is one of the non-territorial adult male, and this is the huge 40,000 square kilometers home range of the Rafi. this individual was also crossing water dams to find the territory and females. The density gave us a result, the highest Eurasian lynx density calculated so far for any Eurasian lynx population, which is 5 individuals per 100 square kilometers. For example, a little comparison in Scandinavia, for example, there are 0.3 lynx individuals per 100 square kilometers, or 0.5 individuals. And then later on, we compare the latitude, density, and home range size for 8 different Eurasian lynx populations, and actually we found out that there was a strong negative correlation between the population density and female home range size given the latitude, but we couldn't find, for example, correlation between the latitude and home range size, which is also an indication of ecosystem productivity. So ecosystem productivity could not tell about the home range size, but rather than that, the density was the main factor driving this. I am passing very fast to the, because I told most of the things related to this. Finally, we had the mitogenome phylogeny of Eurasian lynx in its distribution. There are 6 recognized subspecies of Eurasian lynx. One of them is in our country, and one of them is the Balkanicus. The Balkan lynx is the critically endangered population, which has only 50 individuals in southeastern Europe. And why we cannot see this? Anyways, we use mitogenomes with 2 additional subspecies. One of them was Lynx diniki, and the other one was the Isabellinus, which is the Central Asian Tibetan lynx. We included their mitogenomes inside, we analyzed their mitogenomes, and then we also used previously published mitogenomes from other subspecies, and then we reassessed the mitogenome phylogeny of Eurasian lynx. We had samples from my focal study area, the northeast, and Çıldıkar Research Forest in southwest, and also Giden Gelmez Mountains. And then here you can see the cladogram. We found out 3 main clades, major clades. One of them was clade A, Lynx isabellinus, the Tibetan lynx. This was the oldest matriline in the Eurasian lynx. And actually, previously it was stated that Eurasian lynx actually originated in Central Asia, which was also a good support for this theory, actually. So this was the oldest one, and then the clade B, which is including our lynx population, the Caucasian lynx, and also Balkan lynx is also here inside the clade B. We actually found out, you can see Çıldıkar samples, maybe if you can see it here. It is grouped with the Balkan lynx. So 2 samples we got from Çıldıkar Research Forest actually were related to the Balkan lynx. So we found also evidence of Balkan lynx matriline still being present in Turkey. And probably in LGM, there was a population, individual exchange between the populations in Turkey and Balkans. And it also exists in Turkey until now. When you see actually the other lynx populations in Turkey, they are 50,000 years ago separated from other Balkan haplotypes, let's say. But 20,000 years ago, the Balkan matriline in Turkey and Balkan matriline in Balkans, they were separated from each other and isolated. So up until 20,000 years ago, these populations were connected, or at least connected once and then isolated. So we have the remnant Balkan matriline in Turkey. Here you can see that there was individual exchange between the populations. As I said, we had 6 different haplotypes in Turkey. So all samples were represented by a single different haplotype. For example, we had, although in the Balkans, there was a single haplotype from 3 different individuals, from southeast Western Turkey, we had 2 different haplotypes from a stone study area. And also these are the 4 samples from different areas in Turkey. So each sample represented a different haplotype. And in the Eurasian lynx, actually, there is a high rate of mutation accumulation among different lynx populations. And one more thing to say, actually, all other lineages under clade C, they were relatively younger when compared to clade A and clade B. So Isabelline, the Turkestan lynx, the Tibetan lynx was the oldest, followed by Balkan and Caucasian lynx. And later on, the northern lynx populations were the most youngest ones, let's say. And probably the dispersion route was from Central Asia to Anatolia and Balkans. And later on, Balkans crossed back to Anatolia. And other lynx populations in Europe, they were using the northern route to disperse. I think all of these things mentioned here, I already mentioned them while talking. So Anatolia is a hotspot of diversity and evolutionary pathways, let's say. This study also supported that. We said this, southern Palearctic populations, yes, we already talked about that. And this is also a good indication, maybe now we will have a collaborative project with Balkan lynx people to save critically endangered Balkan lynx, because lately the studies show that actually there are very few individuals, but also in last several years, there is also very low rate of breeding inside the populations. So there is a evidence of inbreeding and they cannot reproduce anymore. So there is a new paper going to come out where we compare their ecology, ecology of lynx in Turkey, lynx in Balkans, and lynx in Carpathians, and also their habitat, for example, preferences, prey preferences, and everything, so that we can prepare maybe a plan to support the Balkan lynx population in Balkans by populations from southwestern Turkey as well. Yeah, I already talked too much, I think. This study in total, Anatolian lynx project led to many new findings for Eurasian lynx. And we are already, European lynx researchers, they were a bit feeling in the beginning a bit threatened because we are changing Eurasian lynx ecology a lot. But now they're accepting it, and we are doing some collaborative projects as well. So kind of we are reshaping Eurasian lynx ecology and genetics. Yeah, that's all. Thank you for listening, and thank you to my all co-authors and collaborators. It was also supported by many institutions, international and in Turkey. And the best thing going to my friend Gürbüz for helping me throughout this project and actually also in my PhD study. Thank you for listening.